Hi, my name is Joe Barco. I'm Senior Director of Workflow Marketing at Unchained Labs. My presentation today is entitled Complete Particle Characterization When You Need It the Most. This is a website I hope you never find yourself on, which is the FDA product recall site. Every time a drug is released and has a recall due to the presence of particulates or other problems, it ends up on this page. This is just a sampling from the first half of this year. These are drugs that have been released and then recalled, all due to the presence of particulates or the potential for particulates, visible matter to show up. Particles can, of course, come in many forms. They could be the drug itself. They can be parts of the formulation, such as an excipient. They could be different forms of the drug, such as a polymorph. It could be raw material that has kind of slipped through. And of course, there's problems in particles that can come from contamination, either from people handling the sample or from the manufacturing equipment itself. Particle characterization doesn't start in manufacturing and contamination, of course. Particle characterization starts at the time of formulation and drug development. So in those cases, you're trying to see if the presence of an aggregate or particulates give insight into the developability and stability of a molecule for long-term development. Over time, that need for particle characterization starts to focus on morphology, which is to be able to characterize particles and to some degree identify them based on the morphological characteristics. As it gets further in process development, manufacturing, and drug release, it's much more focused on identification. And so that identification needs to be certain because in most cases, a particle showing up is an insight into a process failure, which needs to be corrected immediately for the drug to be released. Particle analysis characterization, even though the qualities change over time, have basic needs, just the functionality, be able to count and size particles. Over time, the needs for morphology and identification increase, and they need to become much more precise and much more quantitative. Throughput is always important for any particle characterization, whether that's a ton of samples at once, or whether that's just a handful of samples that have a lot of interrogation that needs to occur, where those particles need to be sort of walked away. Ease of use, always important for any identification or instrumentation type. Minimum sample prep, in this case if you have a lot of samples, it's important to minimize sample prep just because of the hassle of doing so many samples. Later stages, there's always risk that you lose the particle as part of the sample prep process itself. So making that as simple and straightforward as possible is critical. Also important, especially in later stages, is that you have flexibility in sample forms. So not every particle is going to show up in a solution. So you need some way to interrogate things that are not a solution, such as like a particle sitting in a lyo cake or a particle sitting on the side of a piece of glass. So having some flexibility there is critical. Hound is a product that we developed, which is meant to kind of build a bridge between particle analysis, earlier stages of characterization, and later stages where it's much more ID focused. So it's a microscopic based platform that can do count, size, shape, and morphology all at once. It can also do chemical and elemental identification. So particles that are in the solution, and it can do that by Raman or LIBS spectroscopy. For the counting size aspect of it, the solution goes the sample goes into the instrument. It's just lined up either automatically or manually by the customer. It scans, the instrument scans the area of interest, stitches the images together, and then does counting, size, shape, morphology of the particle all at once. So the microscopy-based system can detect particles as small as 2 microns and measure accurately up to 15 millimeters in size. Because earlier stages, you're much more concerned about and more likely to see subvisible particles, and because at later stages, you're more likely to be concerned about visible particles, you can actually count particles and bin them based on any customized bin type that you want. You can have it so that it only looks and counts particles above 50 microns, or you could distinguish in different sizes and morphologies at lower, basically any custom range that you intend. For most of my talk today, because this is meant to be on contamination, I'm going to focus on later stage characteristics of the instrument, which is to focus on the identification aspects of Hound. For identification, Hound uses primarily Raman-based spectroscopy. So Hound can have 2 different Raman lasers in it at 785 and 532 nanometers, and also laser-induced breakdown spectroscopy, or LIBS, which is used a lot for elemental analysis. For Raman, there's two different wavelengths. 785 is the most common Raman wavelength. So it's focused on, and that's because it's a very versatile detection laser. So it can do a lot of different inorganic and organic materials. So most common contaminants are going to come up at 785. Not everything does. And of course, because it's a contaminant, you don't know what you're going to see until you see it. And so 532 is also present in the instrument. And so 532 will be really good at picking up things that might autofluoresce on their own. So that could be a protein, if you're working with protein biologics, drug formulations. It could also be, say, a fiber that might be have a dye on it, which could autofluoresce. And so it can basically pick up things that 785 doesn't give a good spectrum for. So Raman gives a great chemical fingerprint for pretty much any material that you have. And so with two different wavelengths, you have the ability to essentially identify everything. Sample prep for the instrument is straightforward. In addition to what's shown here, you could pluck the particle out of solution, put it on a microscope slide. For smaller vials, you could directly put the vial under the microscope and try to do a detection without removing it from the sample. But in most cases, you would remove it from the sample in some way. So if it's a sample that might have many particles, you can filter it through this gold-coated filter round. The filter rounds have different size cutoffs, so you can remove things that are under a couple microns in size if you want, or retain them on the slide. You can also do, if your particle looks like it's amorphous and you want to measure it in situ, so in solution, so you could put it on the microscope slide on this gold-coated slide, and you can use that and directly cover that up and directly identify from that method. So if you have a protein, it looks like it's going to go through a filter round, silicone oil, things like that, you can measure it by another means. So why use Raman? And the most common mechanism for this is actually to use infrared or IR. The reason for Raman is unique signature. So you can differentiate very highly similar materials. If you look in, do a web search, you can find all sorts of cool applications for Raman, identifying counterfeit drugs, identifying counterfeit money, because the inks will show up in different, and have different Raman signatures. So all sorts of applications like that, you can distinguish proteins from one another, but protein from non-protein as well. One of the big drawbacks of IR is that there's a lot of interference with water. And especially if you're looking at biologics drugs, you're much more likely to have water present. And so Raman will pick up a signature without having interference from water. The reason we use gold-coated filters, as I showed in the previous slide, is because gold is Raman inactive. And so you will get a better signal to noise ratio if you look on that versus looking on something like a nitrocellulose surface, for example, which you would still use in the Hound, but it's not the preferred method. And Raman can pick up particles and identify particles that are smaller, so about 2 microns, which is about an order of magnitude better than what a typical IR instrument will be able to detect and identify. The morphological characteristics kind of come for free as part of the counting and stage of the instrument. So the counting can be done in a few minutes, and reports information on basic information about the particle morphology. So essentially whether it's spherical, whether it's elongated, what's the area, what's the size, whether it looks like it's a fibrous particle. So that will give some additional insight into what the particle type is. And it can also, as I'll show you in the next example, help you classify distinctions between population sizes. So my first example is to talk about a nasal spray. And a nasal spray is a drug where you expect to see particles, expect to have particles. In a typical nasal spray, you have actually an API, which is in a very small percentage, and you have a lot of inert material, in this case cellulose, which happens to be of similar size. So the challenge in say a lot-to-lot release and ensuring consistent product quality, lot quality, is to be able to distinguish how much API is present in comparison to non-active ingredients. And of course, this gets more challenging if the non-active ingredient looks similar to the active pharmaceutical ingredient. So from characterizing the API and the filler material cellulose, you could see that there's a distinction between the two. So the API particles generally smaller, so about 4 microns in size. with some distribution of size itself, where cellulose is a little bit larger, so about 37 microns. But the key thing is that it also has an elongation factor, so it's much more tubular in appearance. So if we look at a distribution of particle types, particles within the sample, you can see that the elongation factor of the API drops off dramatically at about 2.5, whereas cellulose has a wider particle size distribution from about elongation factor from about 1 to 5. So if we're looking at a sample of this type is about 45 to 50,000 particles. That could be challenging to, but you have to identify all 50,000 particles by Raman. And so what we could do is use the elongation factor as a cutoff and say that anything with an elongation factor above 2.5, I don't care about identifying. I just want to identify the stuff below because I'm basically enriching my sample set to ensure that I'm more likely to identify an API that's present. So that was what was done here. So they did a distribution of particles that were under, most of those particles were under 15 microns. So we can see the D90, the distribution of the API was about 12.5 microns. So in this case, we only had to identify about 3,000 particles. Of course, this is all automated and you can walk away and leave the instrument for a few hours or overnight, come back and get the measurements. So that ends up being about 15 times faster to use this cutoff for the elongation factor than it would have been to do the measurements for all samples directly. And you can see from Raman the distinction between a cellulose particle and the graph at the bottom and an API is pretty significant. So they're very, it's very cut and dry about what's an API and what's cellulose by Raman. Most of what I'm going to talk about is about contamination. And so when you have a contamination event, you have to identify not just what is it, but where did it come from? So it shouldn't be present, and a drug manufacturing process can go through a number of different people, a number of different steps, and in some cases, a number of different sites. And so identifying not only what the particle is, but where it came from is critical, right? So you can't release the batch if you've identified visible particles. The requirement is that the drug should be essentially free of visible particles. So identifying a common contamination issue is critical for drug release and to prevent a product recall from occurring later on. So in this example, you have a drug and the fiber started showing up in the drug. So it's pretty obvious it's a fiber, but just by the size and shape. And it's very quick to identify and characterize it as cellulose. So the cellulose peak at about 1100, 1300 is pretty common for most cellulose types. What's not common is this peak that's around 1600 wave numbers, which is unique to this particular fiber. So on the, it's cellulose, there's a lot of different types of cellulose that can come into the laboratory. So it's paper towels, it could be, people's clothing. So the final fill lab started to look at where they could potentially be getting fibers. And so they did washes for their stoppers, looked at their clean in place equipment, looked at their tubing. And so they can see that there's fibers that kind of show up everywhere within those things if they look hard enough. But is this the one that's actually in the drug? Well, no. So they can do a spectrum, get Raman spectrum for all of these potential types of cellulose, which is what they did. They did the materials that the sample comes into contact with. They did clothing and lab coats and things from the people would have in the laboratory. They have paper towels that were used in that laboratory. None of it was matching. None of it was having this kind of peak show up at 1600 wave numbers. So then what? The next thing they do is they go back and look at the API manufacturing site. And so they look at where the API is manufactured. They come up with this match, unfortunately shown in yellow here. So maybe not showing up so well on your screen, but they see there's a match, a cellulose match from there. So from that point, it's like, okay, it's coming from the laboratory. What is it? They trace it to eventually is the tanks. And so the tanks are cleaned in between manufacturing runs. The cleaning is completed with a wipe of a paper towel. And that paper towel leaves fibers behind that end up in the next drug, in the next API. And so they've changed their manufacturing process, their cleaning process to remove the fibers, problem goes away. So on the plus side, what they've also done is they've identified a bunch of different types of cellulose. And so the Raman spectra gets saved as part of the instrument. And so the next time a fiber shows up, they have all of these reference materials available for the next time. So this investigation might have taken a few weeks to get back to the source, but the next time a fiber shows up, they'll have information about what it is and where it's coming from in 5 minutes because they're saving the spectrum. Last example here is to talk about laser-induced breakdown spectroscopy, or LIBS. LIBS basically same concept as Raman. The differences are it focuses on elemental analysis, so metals, glass, heavier elements, and so it can look at identify particles as low as 20 microns. It's very simple to operate. You just line up the particle with the laser, fire the laser. It's no settings. It's just 1 second per shot, and you get the intensity point back out. LIBS is most commonly compared to SEM, which is usually used to do metal analysis for contaminants. LIBS's advantage is it's easy to use, and you don't have to do any special sample prep as you would have to do for SEM. It's very sensitive, just like SEM, and it does layer analysis. And so LIBS actually is a somewhat destructive test, so it hits the surface and will leave an ablation, and that ablation could be used you could actually continue to hit that ablation site. And so if your particle has dirt or rust or something like that on it, you can kind of go down below it and get a more definite ID of the particle and potentially have a better match from that. The instrument comes with reference spectra, and so aluminum is a common metal, of course, and so that shows up in manufacturing, and so that is a reference material. So we can compare that to other sources of aluminum, such as this Wheaton crimp cap. And so the Wheaton crimp cap, if we have a particle of piece of that in a fragment of that in the solution, we can pick that up by LIBS, and it will do a match with the aluminum. But it will also do, it could also do a more tighter match if we're, again, concerned about not just what it is, but where it's coming from. And so I'll draw your attention to the shoulder of this peak at 400 wavelength. And so you can see when it's expanded, the reference in blue is the aluminum standard, and that does not have this peak that's showing up at about 403 nanometers. However, the Wheaton crimp cap does have that peak, as you can see on the right-hand side. And so what that does is it gives us a slightly higher match, and we're more likely to find the exact, identify the exact source of the aluminum contamination. So you can save this as a reference material, and so the next time it's showing up, it's there and it's available to use. The database itself starts with about 150 spectra from Raman and each wavelength, and 50 from LIBS. This is expandable because everyone's process is different with different material types, and so an end user could proactively gather materials that are within their laboratory that are a potential source of contamination, create reference spectra for that, or they can kind of do it ad hoc as contaminants or potential contaminants are identified. And so it takes about 15 minutes to make a reference sample from a material that's put into Hound, and then it's there. You can also export and import spectra. So if you have multiple manufacturing sites and you find common contaminants or find contaminants in one, you could transfer it to the other very quickly so that they essentially learn from the previous sets of events. So in the brief time today, I hope I've given you some indication about what Hound can be used for. So it can be used in a wide range of particle analysis applications. So from simple things like API distribution, where you're more focused on count, size, and shape, and count, shape, morphology, to things like process monitoring, batch release, where you may be concerned about those types of things, but you're also much more concerned with identification that you would need for a particular sample, basically anything that could potentially be showing up in your sample. Hound itself is 21 CFR, you can be used in a 21 CFR compliant manner. So you have flexibility in sample types, all of this capability in terms that you have for a microscope, and the Raman identification capabilities all built in one. You can do this manually if you only have a single particle that shows up in a solution, or you could do automated where you can run multiple samples or maybe hundreds of particles and identify hundreds of particles without actually being in front of the instrument. For more information on Hound, go to our website, unchainedlabs.com, or send me an email at joe.barco@unchainedlabs.com. I'm also available for questions in the roundtable that's coming up. Thank you for your attention.